Welcome back. So last time we looked at the structure and a little bit of the function of the Golgi, and now I want to get more into the details of primarily the ER in glycosylation. And I'm keeping this very general. I'm not discriminate between the ER and the Golgi, so I'm just talking about glycosylation in general and how that happens, and then some functions of glycosylation in terms of protein quality control during co-translation into the endoplasmic reticulum. So as you'll be familiar with, glycosylation is the addition of sugars to proteins. This happens in both the ER and the Golgi. The thing we're going to introduce here to start with is the idea that there are two types of glycosylation, and those are called N-linked glycosylation and O-linked glycosylation. So the N-linked form of glycosylation you can see here on the right-hand side of the screen. This is when the sugars, the oligosaccharides, and that can be any variety of sugars and chains of sugars, are added to the nitrogen of an asparagine residue in the protein. So here is the amino terminal amino group. There's the alpha carbon, and that would be the remainder of the carboxy terminal carboxylic acid group. And so here is the R group, and what we're doing is we're adding the sugars here to this nitrogen in the asparagine. And we contrast that with what we see in O-linked glycosylation on the left here. Here we're going to add our sugars, our carbohydrates, to either a serine or a threonine residue. And so we're going to do a condensation here where we're going to basically link the sugars to the amino acid in the protein via this oxygen. And so this is called an O-linked glycosylation, and this is an N-linked glycosylation. So N-linked is going to be on asparagines, and O-linked is going to be on serine or threonine. So let's take a quick look at the process of glycosylation, and this is complicated, but we can break it down into really into 2 or 3 steps, and whether you break it into 2 or 3 just depends on whether you want to deal with 2 or 3. So I'll talk it through, and then we can look at the 2 or 3 main steps. So it looks complicated, it looks like there's a lot of stuff going on here, and there is, but we can make some generalizations and we can do some learning tricks where we break it down into some steps. So let's look at the sugars we're dealing with: N-acetylglucosamine, mannose, and glucose. So those are the common carbohydrates used in glycosylation. So the whole process starts with the insertion of a molecule called dolichol phosphate into the membrane of the endoplasmic reticulum. So here's the lumen of the endoplasmic reticulum, and here's the cytosol of the endoplasmic reticulum. So dolichol phosphate, like its name suggests, has a phosphate added to it. And the first thing we're going to do is we're going to use an activated N-acetylglucosamine, and we're going to add that along with the phosphate to the dolichol phosphate. So we start out with the UDP, uridine diphosphate, and there is our N-acetylglucosamine, that red square. And so we're going to take the red square and one of the phosphates from UDP to make UMP, monophosphate. And so we now have dolichol phosphate plus another phosphate plus the N-acetylglucosamine. Then we're going to take another activated N-acetylglucosamine and add that, but we're not going to take off a phosphate from the UDP, so we're just going to take the N-acetylglucosamine. Then we can add on any number of mannoses, for example. And at that point, once we built this base oligosaccharide, is what we're going to do is we're going to flip the whole structure across the ER lumen, across the ER membrane, sorry, into the lumen. So I think about this as the first step. If you want to think about this as the first set of steps and break that down in the first part of the process, that's building the base oligosaccharide. And so dolichol phosphate, adding N-acetylglucosamine and another phosphate, add another N-acetylglucosamine from the activated carrier, and then add on some mannose, and then flip. So from the flipping point, everything happens inside the ER lumen. So this is where you can either think about 2 or 3 steps. So let's just take it as 2 steps to start with and think about how that works in my mind. The second set of steps, or the second part of this process, is that we add, for example, more mannoses to dolichol phosphates on the outside of the membrane. So here we're going to add some mannose to the dolichol phosphate outside the membrane. We flip the dolichol phosphate with the mannose inside the membrane, so it faces the inside of the ER lumen. And let's just say we do that 4 times, and that means we can add on 4 mannose sugars to our base oligosaccharide. Now we can add on then other sugars. And so this step here is just a variation of these steps in the middle where we're adding on mannoses. We can also add on glucoses. And so here, rather than using GDP as the carrier, we're going to use UDP. And so we're going to transfer on UDP with glucose, and on goes the glucose onto the dolichol phosphate. Dolichol phosphate gets flipped, and then we transfer that glucose from the dolichol phosphate molecule onto the growing chain of oligosaccharides. And so this process here on the right is the same as this process in the middle, except we're using a different carrier for the sugar and a different sugar. And so this is just a variation of this. So that's why I say that could be 2 steps, or you could think about it as 3 steps, or 2 parts of the process, or 3 parts of the process. When you're building this sugar, it's often being added to the target protein at the same time. So this event of building the oligosaccharide onto the protein, building the oligosaccharide and folding the protein often happens at the same time. So that's often called co-translational glycosylation, and it's believed to help stimulate proper protein folding. So that's a little bit more about glycosylation. Now, the glycosylation, we can obviously think about the roles of glycosylation as being when that protein is presented on the surface of the cell as a transmembrane protein, that glycosylation, those sugars, act as information to neighboring cells and to circulating macrophages and those kinds of things. So we can think about function in that respect. But there's also a role of glycosylation in quality control of proteins translated into the endoplasmic reticulum. And this isn't described particularly well in your book, so I've got some different art, like that last slide, a little bit of different art. So it's been proposed that the terminal residue of a glycosylated protein, so that's the terminal sugars on a glycosylated protein, act as this kind of sophisticated timing mechanism for protein folding in the ER. And so it's a kind of quality control mechanism. And this is a little bit kind of fussy to understand and to look at, because again, there are some cycles that repeat here. So what we have is a group of enzymes here, glucosidase 1 and 2, which can remove these terminal glucose residues from newly synthesized and glycosylated proteins. And so if the protein is misfolded here, and that's shown by this kind of squiggly protein down here, we take off these N-terminal glucoses, and then an enzyme called UDP glucosyl transferase can add sugar back on, so it can add a glucose back on. When that single glucose gets added back on, then these proteins here, calreticulin and calnexin, can recognize that protein because of its single glucose that was added, and they're chaperones, and those are proteins which are able to try and fold misfolded proteins. And so these proteins try and refold the misfolded protein, and then this enzyme glucosidase 2 takes off the glucose, and if the protein is correctly folded, all well and good, and it goes off and it gets packaged into a vesicle, sent to the Golgi, whatever. If we find that this protein isn't correctly folded, then UDP glucosyl transferase adds the sugar back on, and calreticulin and calnexin tries again. So there's this cycling of adding a sugar as a signal to try and get the folding correct, then taking the sugar off, checking if the protein's folded, and if it's not folded, trying again. Now, if that goes on long enough, if this unfolded protein persists in the endoplasmic reticulum, is what happens is this enzyme here, after a certain amount of time, manosidase 1, it will cut off some of these mannose residues, and that then marks the protein for export out of the ER into the cytoplasm, where this proteasome, this big kind of machine, this molecular protein machine, can degrade the protein back down to amino acids and individual sugars, and things can start over. So just to summarize here, these terminal sugars here mark proteins for being checked by this folding machinery, and so they get chopped off and the protein gets checked to see if it's folded, and then a sugar gets added. We check the folding. If the folding's good, off we go and everything's dandy. If the folding's not okay, we add a sugar on and we check the folding again, take the sugar off, and if it's not correct, then we go around again. And then at some point we decide, okay, enough is enough, and manosidase 1 cuts off some of the mannose sugars in the oligosaccharide, and the proteasome in the cytoplasm degrades the protein. So another function of glycosylation is to stimulate protein folding and to act as a quality control mechanism to ensure that proteins produced in the endoplasmic reticulum are correctly folded before they go on to their final destination, or they're used in another process, or they're used to build something more complex. So that's all we're going to talk about in terms of glycosylation. I'll be back next time and we're going to look at protein trafficking, probably one of the most important roles of the ER and the Golgi. I'll see you soon.